This talk has two parts. And let me have my clicker. The first part, I'll go over some recent research where we developed a Ford model for high-level visual cortex. And then the second part, I'll step back and describe briefly how I think about developing quantitative models using neuroscience measurements. These parts correspond to two papers that we published recently. I encourage you to check these out if you're interested in the details. So over the last years, not as many, some years, I'm getting older, I spent a long time developing these Ford models, also known as encoding models for that kind of field, the cognitive neuroscience people, using fMRI data. And I started off in early areas, kind of what you saw in Noah's talk, moved on to intermediate areas, also in Noah's talk, and then most lately have been tackling high-level visual areas, such as the ones you see here. This is a ventral view of the left hemisphere of a subject, and in red are cortical regions that have selectivity for faces, known as fusiform face area, and in blue are cortical regions selected for words, known as visual word forming area. So we're interested in these areas, and these areas are heavily studied by other labs, and we've learned a lot from these other types of studies, but our motivation here was to develop a quantitative understanding, and by that I mean a Ford model that can precisely and quantitatively predict cortical responses observed in these regions. So to do that, we have to start off with good data, and we thought about the problem a little bit, and we designed an experiment. It looks like this. We went off and measured fMRI responses in these regions to a wide range of carefully controlled stimuli, including different stimulus categories, such as words and faces, as well as manipulations of simple properties like contrast and phase coherence, because that might give us some insight as to what these areas are doing. In addition to manipulating the stimulus, which is shown here, you can watch it, we're also going to manipulate the task performed by the subject, because the cortical responses might depend on the task, and we would like to know if they do. In this movie, this is an actual movie of what you would see if you were a subject in the scanner. These stimuli here come in 4-second trials. Each trial consists of 4 images at a rate of 1 image per second. Notice there's a small fixation dot that's rapidly changing its color. All of these stimuli were presented at the center of the screen and were fairly small to simulate what words would actually be in real life. Okay, so just to summarize the experiment, 24 different stimuli, 3 different tasks. So I'm going to spend a minute just talking about the tasks. So if you were a subject and staring at the stimulus, you are instructed to perform one of 3 tasks. These were done in different runs. The first task we call the fixation task. So you're sitting there and you're staring at the dot, and you're going to report whenever that dot turns red. And given how fast the dot is changing and how small the dot is, this is actually a pretty difficult task. In a different run, we would ask subjects to perform a categorization task. So you're supposed to stare at the dot, but now you're attending to the underlying stimulus and specifically performing a 3-way judgment. So you have 3 buttons, and you're just going to report, do you think it's a word, do you think it's a face, or do you think it's neither? And the third task we designed for this experiment was a 1-back task. So again, you attend to the stimulus, but now you perform a different cognitive task. You report with your button box, is the current stimulus the same as the previous one? And importantly, we kept the physical stimuli exactly the same across all 3 tasks. So before getting into this whole Ford modeling business, let's just start off by looking at the data. In this figure, I'm plotting on the y-axis the evoked response we observe in the word selective area, VWFA. And the x-axis here shows you one of the stimulus manipulations, contrast. So here is a measured contrast response function, in a sense, using a face image, a word image, and a noise image. And I'm showing here just the mean across subjects and a 68% confidence interval on the mean. Now, if the task does, I should mention this was recorded under one of the tasks. Subjects were engaged in this small dot thing. And if the task doesn't matter in this cortical region, you would expect the exact same measurements under the other 2 tasks, but here are the actual data. So let's just spend a second thinking about the data. Well, clearly the stimulus category words are generally evoking larger responses, and this is sensible given past research. Another observation is if you stare at the red data points here, these were manipulations of stimulus contrast, and apparently this cortical region is highly dependent on the stimulus contrast. And this may be somewhat surprising if you had thought this region should be invariant, so to speak, to low-level properties. One more observation is the task effect, and you can see that they're huge, so let's just go through that. So for a low contrast word, 3% contrast, the response is weak. It's about 0.2 bold % signal change during the fixation task. But if you ask the subject to do a one-back task, so presumably they are reading the word, for example, the cortical responses in this region go to 0.8, and that's a massive increase. That's a fourfold increase. So looking at these data as a whole, clearly the precise stimulus matters, for example, contrast, and the task you ask the subject to perform matters. So both of these factors are going to contribute to the responses, and so if we're building a forward model of this region, we have to take both into account, and we have to take both very seriously. So this is a brief talk, and the model is a little complicated, but I want to explain it, so I'm going to try to give you some level of explanation of how the model works. So first, just an overview. So we start with a visual stimulus, and it's an image computable model, it's a general model. And the first part of the model has to do with how that stimulus generates a sensory representation in these high-level areas. So I say here ventral temporal cortex is just an anatomical descriptor. It includes these high-level regions like FFA and VWFA. So that's one component of the model, which I'll go over. Another component of the model has to do with how we believe the brain is using the sensory representation to perform a decision-making process task in intraparietal sulcus, or IPS. That's the second part of the model. And the third part of the model has to do with how the IPS induces some top-down modulation of the sensory responses in these high-level areas. So that's the overview, and hopefully it'll become clear once I walk you through some of the details how this all works. So the first part of the model, we have a stimulus. We want to predict responses in these high-level areas. This actually follows nicely upon the description that Noah laid out, although it's a different picture, but it's the same, almost the same underlying model. So in the model, we just take the incoming stimulus image and apply the V1-like filtering that you already heard about, so a set of local-oriented filters across the image, and then some divisive normalization, and the result at this point is really just a V1-like representation. I like to look at it using this type of image. So each pixel in this little color image corresponds to a different position, and the hue is going to correspond to the dominant orientation. So if you just compare the actual image on the screen and the V1-like representation, that all makes sense. Okay, but the core part of the model now is the second stage. So there's actually two sets of linear, nonlinear operations going on. And the model proposes that what these high-level areas might be doing is simply just one more of these types of stages, basically a linear, nonlinear operation, specifically using the concept of a template, whereby maybe the response in a phase-selective region is computing some sort of template matching on the incoming V1-like representation. So here I plot a image, again in this kind of V1-like space, with a putative face template. And so that operation is a linear operation, it's a dot product, so S dot T. And then just some simple subtractive and divisive normalizations after that. So a very simple equation where S dot T is there, and then there's a subtractive step and a divisive step. And the proposal is that gives you a prediction of what the evoked, bold response averaged across FFA, for example, is. So we can take this idea, which is what I started with, and we can see if it actually can help us understand what these areas are doing. So here are some data points in VWFA and FFA. So each black bar is a different stimulus. This is the group average, error bars across stimuli, or error bars across subjects. And importantly, these data were during the fixation task. So remember, the fixation task, subjects are asked to do this funny dot judgment, and they're sort of diverting their attention away from the stimulus. So we can start off and play the modeling game and use fitting and cross-validation to assess models. So we're going to start with the control model. We call it the category model. It's kind of a qualitative or phenomenological model. And this model says, in VWFA, for example, we expect high responses to words, low responses to everything else. And for FFA, high response to faces, low response to everything else. These lines are slightly wiggly because what you're seeing here is the cross-validated predictions. So we leave out each stimulus, train the parameters of the model, predict the left out stimulus, and do this n time where there are n stimuli. So these lines, these predictions here actually are slightly wiggly because of that cross-validation process. So as you can see, this model explains some of the variance, but it's not super satisfactory, and it's very qualitative and non-computational. So we can compare now the performance of the template model. There's actually just a few free parameters given how constrained this model is, just A, B, and C, as you see up above. And it's not perfect, but it does capture a satisfying amount of variance in the data. And we can look at this as a scatter plot, data versus model prediction. Looks pretty good. And then we can also compare against this other control model. So That was a bit of a whirlwind, but I guess you heard it from Noah. But as I guess Noah makes available, and many of the previous speakers, the code is available online. It's all transparent, it all works. And I was just going to spend maybe 30 seconds, hopefully, just to run through something interactively. So I have a little terminal here. It is MATLAB, it's not Python, but that's not that important, I don't think. Okay, so online we make available code as well as the data set. This might be a little difficult. I was just going to show you that it works. So if I can crane my neck like this, I'm just adding some tools we make available. I load in the data set, and just for sanity checking, we can look at the actual RGB images. These are not actual, the whole retina thing I'll get to later in the talk. But these are images, and you can get this. All right, so we just loaded in some stimuli. We'll do very simple pre-processing, just changing the resolution of the stimuli, changing the range of the pixel intensities, and then we'll just run through the V1 part of the model. So here we're just going to apply this set of V1 local oriented filters with some divisive normalization. My code is not super optimized, so it does take a couple seconds. And what we're going to see, just to sanity check again, is the output of this stage of the model, where you're going to see 8 images, 1 for each of the 8 orientations. And that looks something like this. This was performed to the face image, so this is the horizontal filter output, this is the vertical filter output, here are in-between orientations. So everything is sensible, everything works, and we'll just now continue to the rest of the model. We just have to construct these putative templates, very simple operations, and then basically do a little bit of arithmetic to generate a prediction. So if you notice here, this is the equation that you just saw, basically s dot t, and then a few simple subtractive and divisive steps. So I just ran it through, and now let's just look at the results. This was simulating a face template, and I'll show you the stimuli. It's a little hard to see given how many stimuli there are. Here are the predictions of the model, again using a face template. To make it somewhat simpler, I can just plot the 4 that we were tasked to use in this symposium, and that looks like that. Okay, so full contrast face, full contrast word, 50% phase coherent face, and 50% phase coherent word. These are the predictions. Anyway, so it all works. It's not a perfect model, as I mentioned, but it's something. It's a good starting point, and it's all here. So moving on, let's go back to the data in the study. So all we just did was covered how the stimulus relates to high-level sensory representations. But what about these massive task effects? Something completely different. Well, just looking at the data, we already noticed the responses are higher when the subject is asked to do a cognitive task on the stimulus. But in what way? And we can learn a lot just by looking at the data. So here, the x-axis is the response of the visual word form area, the y-axis is the response of the fusiform face area, and each arrow is just a stimulus. And the color of an arrow corresponds to its general stimulus category. So blue refers to faces, and orange refers to words, and gray is neither of the two. But the most important part of this figure is the tail and head of each arrow. So I'm plotting the tail at the observed measurement during the fixation task. So think of that as kind of the bottom-up sensory evoked response. So the tail is the fixation task, and the arrowhead is the response observed during the one-back task. So subjects are engaged in some sort of cognitive task on the stimulus, and so I'm just replotting the data. And the structure of this plot tells you something about the manifold of the representation, and it tells us actually a lot. It tells us these top-down effects are not simple additive effects. So additive would look something like this, whereby there's a fixed kind of response boost for every single stimulus in each area. So we know it just by eye, it does not look like that. Of course, we can formally test it if we would like. We also know it does look more like a scaling effect. So these arrows emanate from the origin. So this is as if the evoked responses in each area are getting multiplied. We can rule out what we might call area-specific enhancement. So you might think when there's faces on the screen, like these blue arrows, if you are asked to perform a task on the face, you might think, oh, it's only the face selective region that enhances, but that would look something like this. That's not what the data show. So we characterize the general sort of qualitative pattern as stimulus-specific scaling. So it's some sort of scaling effect, but importantly, some stimuli seem to receive more top-down enhancement than others. Okay, So we learned something about the nature of the top-down effects. It would also be nice, if we're trying to build a model, to understand where these effects come from. So long story short, and you can look at the paper for more details, we perform a type of connectivity analysis to identify where the top-down effects appear to be coming from, and it appears to come from the IPS, as you see in this figure. And this receives some independent support from some diffusion measurements that we made. Using diffusion imaging, you can perform tractography, find white matter tracks, and there is a vertical occipital fasciculus, which was rediscovered by Brian and Jason Yeatman. You can check out this paper for more details, but basically there is an anatomical substrate that seems to connect the region in IPS that is imposing these top-down effects with the high-level regions that we're trying to understand, such as VWFA. Okay, so now let's put 2 and 2 together. We know it's some sort of scaling effect. It appears to be coming from the IPS, so now we can make a very simple model and try it out. So in this model, we start with the ventral temporal cortex. So this is, again, these high-level areas. What is this bottom-up kind of sensory evoked response in these regions? You can think of it, again, as like a vector in a multidimensional neural space. We want to predict what we observe when subjects perform a cognitive task, and to do that, we take the measurement of activity in the IPS for a given stimulus, and we basically multiply it in. So a very simple equation. So ventral temporal cortex response during the task is just the bottom-up response multiplied by a scaled version of the IPS activity, and that gives us some prediction. So very, very intuitive. Basically, this vector is just getting scaled away from the origin, where the amount of scaling is given by the IPS measurement. So how well does this actually work? Let's look at that. Here's the full set of data from VWFA. We have 3 sets of measurements. Remember, this is the fixation task, then we have the categorization task, and we have the one-back task. We can fit and cross-validate our model, as we typically do, and here is how that looks. Let me just kind of walk you through this. These pink dots show you, in a sense, the bottom-up estimate, so kind of this sensory evoked response. And what's happening is the sensory evoked response is getting multiplied by the IPS measurements, which are shown by these thin lines up here, and the product is shown in red, and that's the prediction of the model. So now we have some sort of prediction for these top-down modulated sensory responses, and it looks pretty good. And of course, you can plot it as a scatter plot, which many people like to do, but I like to look at data, but you can do that. And so we're happy. Okay, so the third part of the model, well, actually, let me review, just because there's a lot going on. We've dealt with the kind of bottom-up sensory sort of feed-forward representation stuff. That was the template stuff. And we've dealt with, just now, thinking about why these responses are top-down modulated from the IPS. But what we don't know yet, or we haven't dealt with, is why is the IPS doing what it's doing? Our model says something very simple. The IPS does what it does because, in some sense, the IPS is the subject, and we ask the subject to do something. So let me unpack that. So think about the categorization task. It's a 3-alternative decision. The subject is tasked with what is on the screen. So they're deciding, is it a word, is it a face, is it neither? And we're just going to exploit some classic ideas from the decision-making literature, specifically the monkey parietal cortex people like Mike Shadlin, and we're going to try to formalize these ideas that they developed using in electrophysiology and apply it to this data set. So here's how the idea works. We start off with sensory responses in our high-level regions, ventral temporal cortex, and again, you can think of it as a vector. And the idea is that this represents some sort of evidence for one of the 3 categories. Specifically, you can think of this, or mathematically, as taking the incoming sensory stimulus and projecting it onto one of 3 hypothetical category vectors, again, in this kind of multidimensional neural space. And the size of the projection on one of these category vectors is, in some sense, what it forms the basis of your decision. So if it's a high projection on the face vector, presumably you have high evidence that there's a face out there in the world on the screen, so that's going to make you respond face. So that can all be written down very simply, and basically the amount of evidence can be thought of as a drift rate in a kind of evidence accumulation model. And so the proposal is based on the incoming sensory response. You have a certain amount of evidence, you accumulate that over some time up to some threshold, and then you make your response. So in fact, this way of writing it out generates a prediction of reaction time. It really is just a function of the incoming response R the category vectors that we propose V, and then there's just 2 parameters if you work through the math. So that's the formalization. We can look at whether it predicts the reaction times because we have those. So in the scanner, we have reaction times of subjects deciding whether it's a word face or neither. That's shown on the y-axis here. Each black bar is one of a distinct stimulus. And then we can fit and cross-validate our model as usual, and it seems to do a pretty good job. The one thing that's missing here is what's the IPS doing? Well, again, exploiting ideas from the monkey parietal cortex people, the idea is that there are neurons in IPS performing this evidence accumulation, and the longer this evidence accumulation process, the longer aggregate kind of spike count we would observe, and we're measuring fMRI, so presumably we would find this in the amplitude of the bold response as measured in our experiment. So we can plot reaction time for our subjects against the IPS measurements. It's not a super fit. Here we're just using a monotonic function to propose some sort of relationship. There are a few data points that don't seem well captured, but this was our first foray into this, and we'd be happy to keep improving on this. So just to put it all together, in terms of the Ford model, we dealt with a template model to describe how high-level areas are representing the stimulus. We dealt with how this representation seems to be used for a decision happening in IPS, and we dealt with how we believe IPS is back projecting on visual cortex, ventral visual cortex, to induce some top-down modulation process. Okay, so that was a lot of detail. Let's step back and think more generally about models. So the name of the symposium is Ford models. So what are Ford models? I guess my reading of it is that they're very natural if you're a cognitive neuroscientist. So at least under my definition, cognitive neuroscience as a field is trying to link properties of some sort of cognitive experiment. So mental operations that our brains perform, trying to link those to neural activity measurements. And the reason we can do this in humans nowadays is because we have good tools for measuring neural activity. So if that's the definition, then I think Ford models follow very naturally. So let me give you a little cartoon that comes from this little paper that I wrote. So all of us, vision scientists, not even vision people, everyone doing experiments with, say, at least humans, conform to this very general setup. So we stick a subject, an observer. The subject has eyeballs. And we construct some sort of stimulus that we're interested in. And there's obviously infinitely many stimuli and many different types of stimuli. But there's some stimulus you typically have to tell the subject to do something. Although some people who do neuroscience, I am a culprit. My first experiment, I didn't tell the subjects anything. I think that was a mistake. But typically we tell the subject to do something and we measure some sort of behavioral response. And if we're neuroscientists, we worry what's going on inside the head. Of course, you can make a lot of progress thinking about the experiment, thinking about the behavior, and making theories about the information processing that people do, right? Visual psychophysics. But as neuroscientists, nowadays we can measure the brain directly, so that hopefully this will give us some insight into the mechanisms that intervene. And we observe some data, and then we simply ask the question, why? Why do we observe it? And I think this really applies to almost everything we do. Why does a specific area light up? Why does the activity vary as a function of this particular stimulus? Why does it vary as a function of task? What have you? And I think models are just a way of formalizing this. So there are many different models we can give of this very simple experiment, which I didn't describe, but basically it's manipulating the visual field position. And models vary in terms of quantitativeness, qualitativeness, detail, and so forth. But these are all kind of descriptions that answer this question. Why is this particular area responding more? So, at least in my view, Ford models is just making this endeavor explicit and quantitative. And I think that's the spirit of this symposium. One implication of the kind of study you just heard about of high-level cortex, if we're building Ford models of these high-level regions, at least, we're going to have to take seriously both the stimulus, which we all do, but also the task. And I think that's just a very simple but important observation. My last slide here is just going into some modeling principles, which I cover in this paper. I like the distinction between functional models and mechanistic models. So let me just give a brief example. The concept of receptive field. So there's some area of this visual field that seems to be important for driving the responses of a neuron or some brain area. That concept is a functional concept because it describes sort of the relationship between stimulus, so region of the visual field and the response, but it doesn't necessarily say anything about the neural circuitry that is actually mechanistically giving rise to that neuron. So of course neurons are interconnected with other neurons and there are many stages of processing, but yet this concept of receptive field is fundamental and really, really useful, and it's a functional concept. So functional models, I think, take us a long way. In terms of evaluating models, I'll just go through this briefly. It was almost a philosophical article, but I believe there's two criteria that we should use. The first one is very straightforward, it's accuracy, although we talked about that a little bit in the previous talks. Does a particular model accurately match some experimental data? Of course, models have different uses, and if you develop a simulation type model that has other, but I guess here I'm talking about models that I develop. Anyway, so accuracy is pretty straightforward as a criterion. The other one is a little more controversial, and it's understanding. Specifically, do we grasp the relationship between the parts of a model, so parameters, computations, architecture, whatever you have, and how that model behaves? And so you saw a little bit of this in the previous talks. Noah twiddled some of the parameters and checked what it did, and that's great because that means we're working towards understanding the model. I guess I should just briefly mention, if you're aware of the rise of deep neural network models, I think you could argue they're pretty accurate on the first criterion, so they satisfy the first criterion, but whether they satisfy the second criterion, I think that's an open question, or rather, I think the answer is no. So let me just end sort of thinking about the content you saw in the first three talks about the retina. There was no mention of the retina at all in the words that I just threw at you over the last 20 minutes. Should we have included the retina? I guess my point of view, I guess I'm pushing into other visual areas, even going beyond visual cortex. I'm sort of scavenging my way, trying to cobble together new model stuff. So I didn't have time to worry about the retina. And I guess, yes, we should have if your goal was to build a mechanistic model. That wasn't really my goal in the studies. It was more to build a functional model. And I guess if you can build functional models that are accurate for the experimental data that you have, I think that's great. And maybe we didn't have to include the retina for our measurements, and arguably there is a little bit of advantage to not include the retina, given that it's complex and it's intricate. And if I would really want to understand the model that I just described to you in these little 3-level components, it helps me understand by reducing complexity, because I can play the game of taking out parameters, switching out components, and seeing whether that matters to match the experimental data that I do have. I'd just like to acknowledge the two people important for the work that you just saw. Kay Yeatman was involved in the study of high-level cortex, and Kevin Weiner, who will be starting a faculty position at Berkeley, was involved in developing some of the modeling principles that I just got done with. And that's it, and I'd be happy to answer your questions.